Hello everyone, welcome to Biology. Today lecture 5 part 10. So my dear student, we are learning post-fertilization events of sexual reproduction in flowering plants. So last class we have learned one of the event of this post-fertilization, that is development of endosperm. Today we will learn about development of embryo. So embryo develops at the microfilar end of the embryo sac where the zygote is situated. So, from zygote, what will develop? Embryo. And this development process will take place in microfilar end of embryo sac. The early stage of development are similar in both monocotyledons and dicotyledons. So the early stage of development of embryo are similar in both monocotyledon as well as dicotyledon. So my dear student, we know that angiosperm or flowering plants consist of two classes. One is monocotyledon, another one is dicotyledon. So in both the class, what happened? The early stage of development are similar. So now let us see that a typical dicotyledonous embryo structure. About its structure, now let us learn. So here is a diagram which shows a typical dicotyledonous embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. So you may see in the diagram, two cotyledons are level and an embryonal axis is there. Okay. Epicotyle is the portion of embryonal axis above the level of cotyledons which terminates with plumule or stem tip. So here you may see that plumule is marked. So plumule is the terminal portion of embryonal axis. Okay. And this plumule is also known as stem tip. So embryonal axis. So a typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. Okay. And epicoty is the portion of embryonal axis above the level of cotyledon. Above the level of cotyledon, you may see which terminates with plumule. Okay. And plumule is also known as stem tip. Hypocotyle is the cylindrical portion below the level of cotyledons which terminates at its lower end in the radicle or root tip. So you may see that the cylindrical portion, here it is level, that is hypocotyle, which terminates at its lower end in the radicle. So radicle you may see, and radicle is also known as root tip. The root tip is covered with a root cap. So you may see that the root tip is covered with root cap. So radicle is covered with root cap. So this is a short description about a typical dicotyledonous embryo. A dicotyledon embryo, this is a brief description of its structure. Okay. Now let us see monocotyledonous embryo. So here is a diagram of it. In grass family, cotyledon is called scutellum. So in grass family, that is grass family, most of the grass are of monocotyledonous in nature, and in that grass family, cotyledon is called scutellum. Only one cotyledon is present in embryos of monocotyledons. So in monocotyledon, only one cotyledon is present in the embryo. Okay. The embryonal axis at its lower end has radicle and root cap. Okay. So here you may see radicle, it is leveled in the diagram, and root cap. So the embryonal axis, okay, at its lower end has radicle and a root cap, and root cap is enclosed in an undifferentiated sheet called collaza. Okay, can you see here? Collaza. So here some undifferentiated sheet, or we can say some cells are there undifferentiated, and this root cap is covered with that undifferentiated sheet which is known as colossia. Then the portion of embryo axis above the level of attachment of scutellum is the epicoty. So above the attachment of scutellum, so you may see the above the attachment of scutellum, here the scutellum is attached, above that level it is epicoty. Epicotyl has a shoot apex and a few leaf primordia enclosed in a hollow polar structure called coleoptile. So here you may see coleoptile, it is level, and this epicoty has a shoot apex and a few leaf primordia which is enclosed in a hollow polar structure, and that hollow polar structure is called coleoptile. So this is a brief description about monocotyledonous embryo. Okay, so this is a structure of monocotyledon embryo. Okay, so I hope that you have understood about the structure of monocotyledon embryo and dicotyledon embryo. So this is how monocotyledon and dicotyledon embryo are developed. So thank you and have a good day.